Uh, all right, so I think what we'll do is let y'all. I want you to write on your slides. Uh, you've only got the only thing that's on your slide that's in red, and this is should be slide page 11 of your slide, the second slide on your set, page 11. The only thing on red on your slide should be this expression right here, if they are on separate chromosomes. I want you to take the time to annotate and put this other red information on there, and I will talk about it. Okay? So put the 9 out of 16, not 3 out of 4 on there. Put the rule 2 to the nth power where n equal number of heterozygous pairs of chromosomes. And I don't have the whole word thing of heterozygous. Where n equal the number of heterozygous pairs of chromosomes. And then the next slide. So now we'll go back, and this is about where, all right. Mendel's law of independent assortment would not work if they were linked, if the genes were linked on the same chromosome like they are here. They would not work if they're like here on the right. The law of independent assortment would not work if the alleles were on the same homologous pair. It'd have to be on different homologous pairs where we have them like they are here on separate homologous chromosomes. So the dependent assortment won't do what needs to be done. It has to be independent assortment. So this has to do with it on separate chromosomes. If in our example, the F1 offspring would produce yellow round seeds, 9 out of 16, not 3 out of 4. See, 3 out of 4 would be yellow and round, and then 1 wrinkle green. That's not the case. It's going to be a ratio of 9 out of 16. And this, now, all right, this is, all right, we're going to talk about this in a minute, but there's a little glitch here that I'll try to clear up. But let me get this annotation bar out of the way. It is just, all right, now. Okay, this really is a dihybrid cross, and unlike what I was saying before, I was ahead of myself. In a dihybrid cross, there will be a phenotypic ratio of 9 to 3 to 3 to 1. All right, and that and there we go. 9 yellow round, 3 green round, 3 yellow wrinkle, and 1 green wrinkle. Now, this next slide has a little glitch. It's not same concept, different traits. Like I said, it's actually the same concept with synonyms. All right, the colors are okay for the symbolism. Okay, it's yellow, green is like that's okay. But what's wrong with it is it's smooth and round are synonyms. Okay, on this one, it's about round and wrinkles. Okay, round and wrinkle shape. Here it's smooth and wrinkled. So the only difference is the synonym smooth versus round. So smooth and round are the same thing. Bottom line is what you see here in the summary is the same as what you see here in this graphic. There are 9 smooth yellow. 1, 2, 3, 4, 5. This is the results of the Punnett square. That's 5, 6, 7, 8, 9 smooth what? Yellow. All right. And it says here there's green round and yellow wrinkle 3 and 3, and that's true too here. Okay, here's green and smooth. That's not right. Yeah, smooth and how can that be? Oh, all right, green. Yeah, green and smooth. 1, 2, 3, and then wrinkle yellow 1, 2, 3, and then the last, the 9 to 3 to 3 to 1, there's the 1. It is homozygous recessive in both, which is what you see is what you get, a wrinkled green seed. Let me drink something here. So what you see here is the same as what's trying to be summarized down here. Now, don't write this down because you'll never get it probably, or you might, but it's the genotypic ratio in a monohybrid cross was 1 to 2 to 1. And the phenotypic was 3 to 1. Well, in a dihybrid cross, the phenotypic is 9 to 3 to 1. The genotypic is 1 to 2 to 2 to 4 to 1 to 2 to 1 to 2 to 1. So, but anyway, that's you can count them up when you look at it. The problem is when I do Punnett squares, I don't like them in diamond view. I like to turn it horizontal, and just that way you can see the male gametes and the female gametes better. This flip-flop side thing looks artistically neat, but it's not good to me to visualize it. I don't think diagonally. All right, now let's see. Yes. Mendel, all right, repeated the dihybrid cross experiment for other P traits and always observed that the typical dihybrid phenotypic ratio of 9 to 3 to 3 to 1 in the F2. Excuse me. Each character appeared to be inherited independently, and his degree of luck is really phenomenal, and we'll talk more about it in a minute. The independent assortment of each pair led him to come up with this law of independent assortment, and that had to be if the genes were on separate chromosomes and not linked. And this other bullet is not as important, but you can read about it later and think about it. But it's if you look at one character, you're going to get the typical unit monohybrid ratio. And I hope you understand that by now that when we deal with hybrid, we say the word hybrid, we're talking mongrel, M-O-N-G-R-E-L. It's something that's not going to breed true, but in life, it may have a better chance of surviving than something that's homozygous. That's why inbred families get in trouble, and that's why breeding dogs get in, and other animals get in trouble when you try to get something homozygous dominant or homozygous recessive, because you don't have much variability. And mongrels, hybrids always have a better chance, almost always. And now, all right, Mendel came up with some other things in the process that he didn't know the concept of gene. He called them factors. That's why I've got it here. Traits are passed from parents taught. These are the conclusions that he came to, other conclusions besides what I've talked about, the two laws. The third law was the law of dominance, and we'll talk about that in a second. But the first is a relevation. The second is a relevation. These genes are factors that exist in more than one form. That's where the concept of allele eventually came up. He didn't know what allele was, but he recognized it was a different factor. And there are at least two different alleles or forms for each trait, which accounts for variation in inherited characters. And there can be more than two forms, but and then the law of dominance is he found that's where you come up with purple or white and not blended inheritance of pink, and that's so his experiment turned out to be one of another aspect of luck is complete dominance. Each gene has two alleles, one of which is completely dominant on the other, and thus not an example of incomplete dominance, not blended inheritance, not pink. Which actually does occur in other species, different traits. So he was lucky, and you know what? He was really lucky. Can you think about the fact that Pisum sativa, garden pea, has an N number of 7 and a 2N number of 14. So he had of the 7 chromosomes, he had picked traits that are on all separately on the 7. That what kind of odds are that? But And so he was lucky in that regard. He was lucky that each character is controlled by a single gene, a single factor inheritance. And he was lucky that he got complete dominant situation and not blended inheritance. Now, beyond Mendel, though, and more realistic and complicated scenarios, I'm going to talk about them. We'll talk about incomplete dominance, blended inheritance. We'll talk about polygenic inheritance in a few minutes. We'll talk about pleiotropy in a few minutes. I'm going to talk about epistasis on this slide, and I'm not going to talk about the other mechanisms, but I just want to assure you there are more, and more than we have time to go into in further in genetic studies. But that's why a general botany class would talk about a little bit of genetics, and the genetics class would spend more time on these topics. But anyway, let's go ahead and talk about epistasis now. In genetics, epistasis is a phenomenon where the effects of one gene are modified by one or more other genes. And sometimes these are called modifier genes. Epi means above and hypo means below, but the one that does the controlling is called the epistatic gene, and the ones that are suppressed are hypostatic. But so what happens is a pair of genes modify the expression of another pair of genes, moderate it. And that was not something that Mendel dealt with, and that's probably good because it would have confused him. But we're going to talk about these others right now. And first, I've actually already blurted out when I said blended inheritance. Okay, snapdragons can have red, pink, and white. That's incomplete dominance or blended inheritance in snapdragons. This is what it looks like. The phenotypic and genotypic ratios are the same, 1 to 2 to 1. They're identically the same. The blended ones are heterozygotes that are here, and the homozygous dominant there, and the homozygous recessive there. And what you see is what you get in this situation in the F2 down here. F is for filial, which has to do with son and daughter. But now here it goes incarnations, the same thing. Red, pink, and white. Now, polygenic inheritance is in polygenic inheritance. A good example is human skin color, human height's another example. Plant height and tobacco is similar in that it's controlled not by a single pair of genes, but by a series of genes. Assume that there are 3 loci, A, B, and C, each of which has 2 alleles. Imagine the pure breeding short plants are all that and tall plants are that, and anything in between is some variation of that. So the tallest plants would be that, the shortest plants would be all lowercase. And so that's poly means many, and many genes are involved. And height in humans is a best example. Skin color in humans is another. Pleiotropy. Read this first paragraph on your slide. I want you to go ahead and read that first paragraph on your slide, and I'm going to go, while you're doing that, I'm going to go back and find the slide that we talked about last time here that illustrates that point. Gosh, I thought it was, oh, here it is. All right. All right, flower color and flower position. That's this trait, this trait and this trait are examples of pleiotropy. And let me move back to the slide that we're on. Okay, now, during this study, Mendel made several interesting observations regarding the color of various plant components. He noticed that plants with colored seed coats always had colored flowers, and and colored leaf axils. He also observed that pea flowers with colored seed coats always have white flowers and no pigmentation in their axils, talking about the leaf axils. In other words, in Mendel's pea plants, I guess the axils of the fruit, not the leaves. In other words, in Mendel's pea plants, seed coat color was always associated with a specific color and axil colors. That's what that's trying to say, and that's an example of pleiotropy. And we now know that Mendel's observations were a result of this phenomenon, which a single gene contributes to multiple appearances, multiple phenotypic traits. In this case, the seed coat color was not only responsible for seed coat color, but also for flower and axial pigmentation. Now, what happened after Mendel is what we call something I actually introduced in the very beginning. I brought it up, the modern synthesis. The final word on genetics and evolution. This is what happened when Mendel's studies got resurrected 35 years later. And actually in the 30s, population genetics made its debut and got involved. And so after all of it was done, we've got the new version of genetics, the modern synthesis. And the relationship between genetics and evolution and ecology and population genetics and all these other subsciences of biology came into play. And it became a powerful set of concepts. Good thing Darwin, he would have been a good thing Darwin and Mendel didn't, in a way, know about all this. They would have probably blown their minds at that early time. But anyway, that's what the modern synthesis is. Now we're going to move into evolution, and notice that the word evolution is appearing here. And it'd be hard to talk about evolution if you hadn't talked about plant reproduction. That's why we did reproduction, and it's hard to talk about evolution if you don't know anything about genetics, and that's why we just did what we did on genetics. Now, I anticipate that we will, what'll happen today, I suspect in the time I've already almost killed half our time with our tangent, but maybe we could break that up into two different things. I might decide to go in and I can do a little bit of editing, Steve, but not like you do, but I can go in, maybe I'll separate that little tangent I went on and just have, we'll see what I do with it. But today we'll get into pollination mechanisms. If we get into plant breeding systems, I'd be surprised, but that would be, all these last four bullets would be the last presentation we have, which hopefully will be next week, which is the 15th. But so now I already talked about this a little bit. Now I'll say this again because it's so important. Asexual reproduction is not totally bad. It's a short-term advantage and a long-term liability. And a good example of it was what happened in 1846 in Ireland when the potato was a staple of the people in Ireland. The average person would eat 8 pounds a day of potatoes. Can you imagine? Well, they'd put all their genetic eggs in one basket, and when the late blight of potato, Phytophthora infestans fungus, destroyed the crop, the people died and migrated to the U.S. and other places. So now, asexual reproduction, if you think about it, some of the worst weeds in our world, in the plant world, are those that reproduce asexually. Steve and I were the other day talking about Johnson grass, sorghum halepense, and I think in the top, it's rated as one of the top 15 worst weeds in the world. It comes out of the Mediterranean, and it spreads by rhizomes, making it really bad. Another grass in the south that fortunately barely gets into Georgia, or last I looked, was Kogan grass, and it reproduces by rhizomes. And so when you have a plant reproducing sexually and asexually at the same time, you're going to have something that potentially is problematic. Now, this little chamber bitter, I don't think reproduces asexually, but it's bad enough. I can only imagine what would happen if it did. But we're going to concentrate on sexual reproduction. That's our focus here. Okay. Let's see. Now, propagule will be defined on the next slide, but that's we'll get into that. All right, so we'll talk about breeding systems next time. I don't think we'll get much into it, and let's see, and this will be next time. And even though we'll define propagule now, we won't get into the details next time. Now, these are some terms that we need to review or hopefully not introduce, because we've been talking about them, some of them from time to time, except for propagule. That down here, any plant part, asexual or sexual, that comes detached from the parent plant that can form a new plant. That's what a propagule is. In bryophytes, we talk about Jimmy, and we talk about that's an asexual one, and there's brood bodies, which are miniatures of the parent plant. Examples of sexual propagules include the seed and spore. Now, you think about it, that has to be meio, M-E-I-O, myospore, because a mitospore is not a sexual derivative. A myospore is a derivative of meiosis, and so technically you'd have to write the word myo in there because to call it a sexual propagule. Now that's splitting hairs, but that's the truth. Now, I think most of you know already, but you can't get fertilization in plants until you have pollination first. Pollination has to precede fertilization. In pine, that process takes 2 years to get a seed, because the first year involves pollination and the growing of the pollen tube in through the megasporangial wall to reach the micropyle. And one of the reasons that the angiosperms are more successful, they can do that in a year. And actually, some gymnosperms can do it in less than 2 years, too. So now pollination is ultimately the pollination function has to do with bringing the gametes to the same location, so fertilization can take place. That's what pollination is. And in angiosperms, it's transferring from the anther, which is the microsporangium, to the ovule, or actually to the stigma in angiosperms. In gymnosperms, it's to the down in the the cone. As you go down in the bottom of the cone, you have the ovule, the seed sitting down there. The seed is the fertilized ovule, egg that's been fertilized. And so getting the pollen from the male to the female is the bottom line. And in the case of the angiosperms, it's to the stigma, and that's down a style could be as long as a corn silk in corn. But that's what pollination is, getting the sex cells in close proximity so that fertilization can take place, zygote is formed, and the embryo comes from that. Okay, and I already talked about a propagule. Now, we're going to go through examples of pollination by water, wind, and then separate abiotic and biotic. Ecology, we don't have, and I said more than once, we don't have a separate segment of this course. It's called plant ecology, but we'll talk about it in bits and pieces like we're doing right now. Ecology is a study of organisms in their environment, in their habitat, if you will. Part of an organism's environment is abiotic, that's non-living, that's air and water type stuff, and minerals. And part of it is biotic, that means part of it is other organisms having to compete and prey maybe on them sometimes, or be preyed upon. That's the biotic element of the environment. My point is, when you talk about pollination, you can't escape ecology. Pollination by wind is exploited by both angiosperms and gymnosperms. Mostly gymnosperms depend on it, with gymnosperms rarely being insect pollinated. Gymnosperms, in case I forget to say it, there's a few of them are beetle pollinated, and there's some cycads that may be ant pollinated. That's still, the jury's still, that's pretty well a good hypothesis. But by and large, gymnosperms from the Mesozoic, which are mostly extinct now, and we have about 1,000 gymnosperms left and 250,000 angiosperms. They're depending on wind pollination for the most part. And the angiosperms have capitalized on, I'll point out in a minute, about 80% are biotic or pollinated by animals, and 20% are doing like gymnosperms, doing the wind route. And some angiosperms going back to the wind pollinated secondarily evolved back. And examples of that would be willows and oaks and birches. Back to the wind pollinated route. And if you think about it, people, the temperate zone and tropic zone, as you head toward the temperate north, which we're in Georgia, we're south of us is getting tropical, north of us getting temperate. As you head north, you get wind pollinated more so. And when you get in the tropics, it doesn't pay to be wind pollinated. Why? Because if you're trying to get pollen from plant to plant, especially if they're dioecious, separate plants, you have to go through all these foliage of the tropical vegetation, and that's just not this there year, pretty much year round, unless you're in a monsoon situation where you get leaves being lost due to drought. So bottom line is, where do you find most wind pollinated species? The boreal forest of the north and the temperate forest of the northern hardwood forest. Get birches and beeches, and beeches in the oak family, but are more northern, and the more equatorial you go, the less wind pollinated you get. Timothy Estep, our vice president, just got back from Alaska, and the diversity of trees, that is just not much, and doesn't take much to learn the flora up there. And willows, I got a book on trees and shrubs of Alaska, and half of it is, a good bit of it is willows and poplars that are wind pollinated. So tree diversity in Alaska is not so great, not surprising anyway. So any questions at this point? Okay. All right, so let's look at, now actually pollination by water is an angiosperm thing, and there's not many of them among that 250,000 that have that. So let's talk about one of them though. I love this common name, water chariot family, because that's what goes on. Now I'm going to kind of read some of this so I can get it, because it's kind of involved in the next slides. Show. Actually, this slide and this slide are related. Now, Vallisneria americana is commonly known as the American eelgrass, is one of the species in the water chariot family. Now, what you're looking at is a male flower that has reflexed sepals. Two of the sepals are showing, the other sepal on the other side is not showing, and this is a very small flower. And like this slide shows, the plants are dioecious. This is well, if you compare, I wish the artist had put these separated. That's what the actual plant looks like with rhizomatous connections here. But look at the long peduncle that you've got here. And staminate, and that peduncle, that's a fruit. But the pistillate flower, this one over here on the right corresponds to this one over here. And the female has a tremendously long peduncle, and there's the what looks like an inferior ovary. I'm not sure that that's what it's got, but probably is. But anyway, the female flower is sitting up here on the surface of the water, and the male flower becomes separated from this little booger, becomes separated from the from its parent plant down here, the staminate spathe, which is kind of like a jack-in-the-pulpit situation of a spathe. And the male flowers are all stamens, nothing else. and they float up, and it's like a water chariot. It gets borne over to the female, and that's how pollination takes place. The reflex sepals that you see here act like pontoons and just keeping the stamens above the water and the pollen dry. So the flower doesn't really open until it gets up to the water, so you don't get the pollen wet. And so eventually that bumps into one of these and fertilizes that female transferring the pollen over, and then you get fertilization taking place after that. Okay, another example, again, not common in the angiosperms, but I'm giving you some examples. This slide actually was not in my hard copy, but it was actually, no, the previous slide I had to add. But this one was there, and what you've got here in this surf grass, a family that most of you'll never probably see, is Zosteraceae. You got another kind of water pollinated angiosperm, and the pollen looks like is linear pollen grains. And these strands of pollen grains get caught in these styles. It's kind of like the comb, the bristles of a comb, and that's how it gets pollinated. They get hung up in this styles, and the stringy styles catch the pollen, and that's how water pollination takes place in this plant. Now, moving to, let's see if I've got that statistic somewhere. I'll summarize that again, the water, the biotic, abiotic later. But all right, this is in Pennsylvania in about 1979. I took this picture in the mountains of Pennsylvania. Got some dirt on it here on the original slide, but now poplar, quaking aspen is a widely distributed plant, and what you're looking at is the female catkins, because the willow family is dioecious. Both the willows and the poplars are dioecious. And you got this. Notice the background is everything is leafless, and that's what you want. Is you want to get pollen transferred from another plant to these. To this is a catkin or a mint. If we enlarge it, in willow we get that, which is another genus, but same concept. You have staminate flowers on one plant, pistillate flowers on another plant. The staminate plant is not shown in this picture. Now, here's a blow up of what the male and the female flowers look like, with not petals but a bract, which is a modified leaf. And here's a neat thing, people, that willows is one of those that I mentioned that probably secondarily evolved back to wind pollinated, but look what happened. The female keeps a gland that's a nectary type of gland down here retained, and it's sort of I don't know if that's one over here or not, but this one, the bottom line is that there is a potential for insect pollination in this primarily wind-pollinated plant. So this plant is hedging its bets, so to speak, in that regard. Even though this is a tiny structure and there's not the kind of usual attractant that we see, because let's face it, the pistillate flowers in this family are not very showy. And how many insects are going to be running around in February in Pennsylvania? Not many, folks, not many. But we have willows down here, not many, though. As you go to Florida, there's only one. Salix caroliniana, which is on the coast of Georgia. But in fact, there's only 3 willows that are common in the whole state of Georgia, despite numerous species being here. But, so it's really not a formidable genus to identify in this state. But all right, so anyway, talk about a bare bones flower situation. Well, that's what we've got. Now, oaks, this is dioecious in the willows, but oaks are monoecious, where you have we have one house and 2 bedrooms. Here you have 2 bedrooms, and they're in different houses. This has got one house and two bedrooms, meaning different branches. Most of you haven't probably ever seen the pistillate flowers of oak. You'd have to look, even I have to look when I'm looking for them. And three stigmas and showing, and then this is the young fruit, and this is a mature fruit, an acre. But the pollen has to land on these stigmas, and in a typical, it's kind of sad but funny in a way, or funny but which way to go with it. But most of you may not have seen the female, but you've darn sure seen the male all over your car, along with pine pollen too, which is the Mickey Mouse stuff. The pine pollen has little Mickey Mouse ears to help bladders help it get around, but pollen on the oak doesn't. But it's still the kind of pollen that you see in mass. That's all over my porch, and I have to power wash it every year, because each tassel or catkin that you have, now that catkin that runs about 2 or 3 inches long, it corresponds to the willow catkin, but and it's staminate. This is a glass model from the Chicago Field Museum of Natural History that blows it up. But each stamen has about each male flower has about 8 stamens, and there's probably maybe 40 on one tassel, one catkin or a mint, A-M-E-N-T. So it's playing a numbers game. When you, because there's no insect going from point A to point B, you got wind blowing the pollen, and they're going to do it early before everything leaves out, including their own leaves sometimes. These are young leaves. But anyway, there's a glass model of counterpart of that, showing what a female flower looks like, which is an actual photograph, not the best. This showing a little older on myrtle oak, which I planted in my yard as a hedge, and I regret it because I'm having to cut them every month or so. That was a bit, I wanted something natural, and I got it. But anyhow, now, grasses and a lot of the graminoids, like sedges, another graminoid, and to a lesser extent, the Juncaceae, also a graminoid, are wind pollinated. Anemophily, windflowers, anemone in the buttercup family. That's what this means. And an anemometer is a gadget that measures wind. So the flowers are small, numerous, often unisexual, more so in the sedges. Perianth is reduced or absent, or that's the reduced part, not showy. Flowers are often produced in mass, and that's true of these too, of trees. So that's a big group of monocots that is wind pollinated. Now, this sort of summarizes what's going on in the angiosperms, and to a certain extent in gymnosperms, but comparing abiotic and biotic pollination mechanisms. Again, abiotic pollination refers to situations as pollination is governed or mediated without the involvement of organisms. And in contrast, could be insects, could be birds, could be bats, or other animals. Now, in my notes, I've got 2 statistics. The first, it says only 10% of flowering plants are pollinated without animal assistance. In other words, only 10% of flowering plants are considered abiotic. And the wind is the most common, and water is not so common. Then I've got further down, about 80% of plant pollination is biotic. So that doesn't add up. 80 and 10 is 90. Where's the other 10? So I'm thinking that it's probably, we could say the vast majority, more than 3/4 of all plant pollination is biotic, and somewhere between 10 and 15% or so are wind pollinated. And if you want to get higher percentage of wind pollinated, go toward the poles. And more animal pollinated, go toward the equator. Okay. Let's see. Before we leave this slide, of the 20% of abiotic that I thought, well, see, there I go. I get the correction here. About 20% biotic instead of 10. 98% are wind pollinated, 2% are water pollinated. And so that, like I said, the angiosperms, it's not very common. And then in insect pollination is not very common in gymnosperms either. So, all right. Now, before we leave this slide, let's talk about this adaptation to promote pollination. There's something called in gymnosperms a pollination drop, and the pines depend on it. The cycads and ginkgo don't need it because they've got the sperm swim all the way to the egg. But the flagellae are lost on the gymnosperm's pollen, and so a drop of sugary fluid is secreted through the micropyle in gymnosperms, and the windblown or insect-transported pollen, insect in the minority, falls into the pollination drop and is drawn through the micropyle to the ovule as the pollen drop as the sugary fluid drop is reabsorbed. So that droplet gets reabsorbed and brings the pollen with it, and that's what gets the pollen, the male gametophyte, in the final proximity to that egg to fertilize it. Okay. All right. Now, all right. Coevolution is evolutionary links between animals, angiosperms, and animals. One of the reasons there's 250,000 angiosperms and only 1,000 gymnosperms in the Cenozoic, and we're living in the age of angiosperms, which is the age of mammals, as opposed to the Mesozoic, which is the age of the dinosaurs and gymnosperms. One of the reasons that angiosperms have evolved to the extent they have is they've co-evolved with animals. And as we said, the majority of them, of the angiosperms as opposed to gymnosperms, are bodically pollinated. Now, today, in the remainder of the time, we're just going to concentrate on pollination, and transport and dispersal of the seeds is something we'll get into in the last series of slides in the last lecture. So this is what's going on in over here, honeybee. And actually, I think this is, let me see, I think this is, we got cactus in a going on at night. This could be in a saguaro type cactus with bat pollination going on. Now, in this one, we've got flower pollinated by hummingbirds, and they really like red and pink a lot. And so the long, thin beak of the hummingbird, this is a rufous hummingbird, enables the animal to probe into and get the nectar. And before he leaves, though, the anthers dust pollen on its head, and that's where pollination takes place. This situation is a Scottish broom flower, and honeybee is harvesting the pollen and nectar, and the flower has a tripping mechanism that arches the stamens over on the bee and dusts it with pollen. So some of it rubs off on the stigma of the next flower. So that's what's going on in these three slides. This over here is a nocturnal situation, and moths can do the same thing. Thinking back to the introduction in the course, we said that animals are more dependent on plants than plants are dependent on animals. This would be an obvious exception. This is the penalty. There is a penalty, though, for getting specialized, especially you see it in orchids when there's only, you're dependent on one pollinator and something happens to it, then you're in trouble. That's what happens when coevolution can get you in trouble when you put all your eggs in one basket. Now, in the usual attractant for pollinators is the perianth. And it reminds me of a sad story. I taught a workshop one time on plant identification, and the first day I introduced what a perianth was. And the next day I expected people knew what a perianth was, and this question would come, what's a perianth? I almost lost it because a perianth is a collective term for the calyx and corolla. And the calyx is the collective term for the sepals. The corolla is a collective term for the petals. collectively everything that's no spores sporangio on it is called a perianth. The stamens are not sterile. They have sexual structures on them, and the carpels are not sterile. But the sterile part of the flower, if it's there, is called a perianth, and that's the norm. Now, in the situations like the Nyctaginaceae, the 4 o'clock family, there is no petals, but the sepals look beautifully colored. Sometimes there's bracts, like bougainvillea, the tropical vine that has colorful bracts in addition to the colorful sepals. The 4 o'clock outside in the yard there, has no petals at all. The sepals are petaloid. Another good example is clematis, Clematis, in the buttercup family. Buttercup family often has no petals, but sepals are colored. But that's the norm. In some situations, like the myrtle family, which we don't have too much here, the eucalyptus is a big component there in the southern hemisphere, particularly Australia. But we do have legumes like the Mimosoides subfamily where the stamens are the showy part of the flower, and they're the attractors. This is something I've got in my yard, canna, and in canna, the anatomy of the flower is especially peculiar because the stamens are modified. A staminode is a sterile stamen, but most staminodes don't look showy like you see here. You can't hardly tell. It's hard to tell a petal from a staminode in the cannas, but that's the case. That serves as an attractor for pollinators. The crown-like structure in Narcissus is an attractor. And in the case of, I hate this, flowering dogwood. What a stupid name. It implies the other dogwoods don't even have flowers, and they do. They're just not showy. There's a little bunchberry shrubby thing in the same genus, Cornus. But this is the Cornus florida, the so-called flowering dogwood. But these are bracts. Your flower cluster is in the center here, and they're very small 4-petalate flowers. But the attractor here is the bracts. The bract is a showy leaf-like structure associated with a flower, and this certainly fits. Another attractor besides visual cue is an olfactory cue, usually coming from the perianth. It could come from those nectaries, those glands, if they're present. And it could be sweet, oftentimes, or sometimes foul and fly-pollinated flowers. I'll show you one in a minute. But right now, let's talk about the reward trick mechanism that sometimes is present. It can be a nectar, pollen, wax, or oil reward. In this water lily, though, there's some tricking going on. And Dutchman's pipe can trap the insects, but what happens in water lily is a unique kind of pollination strategy. Now, strategy for something that doesn't have a brain, too. On the first day that the water lily flower blooms, its pollen is not yet released, and instead a fluid fills the center of the flower, covering the female parts. All right. Should an insect visit the flower, the design of the petals causes it to fall into the fluid, and the insect, if it doesn't drown, and sometimes they do, the insect is covered in pollen, and the pollen dissolves in the liquid and fertilizes the flower, and that's on another flower. So he gets pollen from one plant and transfers to the next day. The fluid, no fluid is produced, and the pollen is released. Instead, the insect falls into the fluid and emerges unharmed if he doesn't drown. There's a few unlucky ones that do get trapped and drown, but it's really a complicated mechanism in the water lily. Now, entomophilia, as you might expect, is insect pollination. And sometimes nectar guides get involved, which are humans don't always see these. Some of these are color cues that humans can't see. And the actually ultraviolet light is involved in some of this, and nectar guides are like lines that help to guide the bees in. And the this is in the snapdragon family. You can tell that. And this is probably, it could be a delphinium, or it could be a Linarian in the same family as that. I can't tell for sure, but there's a spur, and so, and that's often involved in insect pollination. So bees and butterflies are big pollinators, and then moths. Now this particular flower is a Floridian type thing, moonflower in South Florida, and the thing's about a foot long, and it's mostly at night, and moths get involved. Now we don't have that in Georgia, but we do have something called Ipomea, same genus, macrorhiza, macrohiza. It's not common. I got it in my yard. I've never seen it flower because I just haven't been there at the right time of night to see it. But I've got pictures of it. A friend gave me from up on in Sylvania, and I just haven't put them up on the internet yet. But it's not quite as showy as this, but it's in a way it's just as prettier. But night flowering plant, not your common morning glory that you see in the daytime. Now this is the largest flower in the world. This is Rafflesia is a genus that has flowers that are like, some of them can be 3 feet across, and it's a parasite in Borneo, at least this one is, that has no leaves and no. It's just a root parasite on other plants in Borneo, and the thing can produce up to 4 million seeds. But notice, and this is a xerophytic plant in the milkweed family, or the dogvane family, if you lump it into the Apocynaceae. Stapelia, I don't know which species it is, but they tend to be cactoid-like plants in the sense they're succulent and live in arid habitats. But what do these have in common? This is tropical, this is more desert. They have that fleshy color. Sarco means flesh. And they have that color that would attract flies. And they say you can. I know I smell this, but I haven't been to Borneo and smelled this, or been to a conservatory that has it. But they say you can smell it from a long way, and it smells like rotting flesh. Birds like pink and red colors, and that's what you get in this Erythrina. And bird of paradise is related to banana, and or maybe not in the same family anymore, but Strelitziaceae is not far from the Musaceae, the banana family. But with that, you can see why birds would like that. And then this kangaroo paw that's from Australia, that same family as redroot that's in our flora. And then we have a cannonball tree that's bat pollinated, and a lot of cacti are bat pollinated. So anyhow, so these are just different examples of pollination mechanisms, and that brings us to where we're going to stop for next time, the breeding systems. And I'll go back to show you where, for orientation's sake, where this breeding systems appears on this slide. Okay, right here, breeding systems. So that these bullets here will be dealt with on the last session. And are there any questions? Okay, I hope everybody's still there. I'm going to return to the meeting.